little bit more in biology, but don't be afraid. As last time, it concerns pretty much that you get a feeling of the role of membrane proteins in the functioning of a cell, and I hope that this knowledge will help you also in, for instance, studying biological membranes or in designing membranes for particular applications, for particular purposes. And the topic of today, the first session, would be the origin of membrane bioenergetics. So what we want to understand is, how do membranes help that a cell is supplied with energy, and that's typically in form of ATP. How is ATP generated in a cell, and what is the role of membranes in this process? So let's start maybe to go back in time. We go now very, very long back. Not only 100 years, 1,000 years, milliards of years we go back. At that time, the Earth was basically formed, cold enough that water would condensate, that we would have liquid water. We would still have some volcano activity, and maybe it would look like that. And you see here, for instance, a place a little bit on volcano stone, a little bit with sulfur, and the atmosphere would contain carbon dioxide, water, carbon monoxide, nitrogen, ammoniac. There would be no oxygen. But that was the time when life started. That was it. So all life started actually on Earth without the presence of oxygen. And you can maybe, imagine that these type of rocks, when they are from the volcano, they are very porous, like a sponge. If you have at home maybe a sponge, there's a little channels in. So this material has a lot of cavities, small cavities. And maybe in such type of cavities, the organic material that was there in the ocean started to undergo simple reactions, started maybe to form a layer of lipid, starting to make the first barriers, so that in a cavity you would have a closed space which would almost mimic like now a cell. And that's the basically idea how life might have started in terms of getting separated from the environment and basically to start to use organic material that at that time was very rich. You see also the sky was not really blue, oxygen was not there, so no ozone layer. So it was actually more a type of a sky that you would find maybe on the Venus like, so very cloudy and not so beautiful. When we look now, this morning was beautiful weather, you see a beautiful sky. But life started, and maybe the very first cells that you can think about would have now this type membrane that we discussed, not necessarily a bilayer that would have basically two independent lipid layers, but maybe you would use these type of lipids even that are connected, where the tails are connected, these very strange lipids that we are calling border lipids. And what would need this organism to live? It would need to take up substances and would use these substances to actually generate ATP. And to do so, it probably would also very early need a type of facilitator and uptake. And we talked about that this could be secondary uptake systems that are driven by a gradient. So most likely, these first organisms would have a mechanisms to extrude protons and use this gradient of protons to move organic substance in, to accumulate organic substance. So that could be the first organism that we think. Maybe they even use these proton-driven transport not only to uptake material, but also to actually secrete material like toxins to protect themselves in this ocean around with all these organic substances. Now, when an organism would do this, it would be, of course, very costly. So it would basically take up organic material, would do glycolysis, would use the ATP to create this proton gradient. So it would actually use quite a lot of energy just to take up. So organisms that would basically came up with a system that would not use ATP to create a proton gradient, but would use energy-rich electrons that are also produced during the process of glycolysis, would use these electrons instead. They would have an advantage because they could use the ATP for other processes. So probably another thousands or millions of years later, some organisms came up, or at least one organism came up, with a proton pump that would be not driven by ATP, but would be driven by electrons that are derived from the organic material that they would have taken up. So these organisms would have a huge advantage now, because they would still make a proton gradient that can drive uptake, but would not waste ATP, but rather would use the energy-rich electrons. Okay? And if such a system would be very efficient, so if basically these electron-driven proton pumps would very efficiently use the energy that is delivered from electrons from the organic material that is degraded, you would build up like a cascade of proton-driven pumps, so you would basically be able to make a very strong proton gradient that then could be used not only for the uptake of material, but allow basically the ATPase to work reverse. Remember, we talked about yesterday about ATP-driven pumps. There are 3 types. And there was basically one example, the F-type and the V-type ATPase, where the F-type works reverse. So that would be a reverse-working ATPase that now makes ATP. What a wonderful organism. It can fully use all the energy that are in the nutrients that are taken up to generate a strong proton gradient that can be used to make ATP and on top can be used to take up organic material. So these bacteria, they would best compete with all the other bacteria around. They would basically, that would be a big bang in evolution. Those organisms would now start to occupy the whole ocean. Yeah? And you see, so the principle here is without a membrane, such a system would never have been able to build up in terms of separating the cell from the environment and in terms of basically allowing the cell to drive the metabolism, to generate ATP, to take up material for degradation and for synthesis. Okay? So probably another million years, the world was very happy with these organisms, and what they do? They would not eat up all the organic material. The ocean would be full. But of course, if we consider this, at a certain moment, the organic material would be then limited. No? So these organisms would start to compete for the organic material, and that gives a lot of pressure for organisms that maybe find now a new solution to actually generate ATP. And what happened was that basically with the purple bacteria, organisms appeared that can use the energy of light. That was, again, a major development. But the prerequisite was that they have membranes to be separated from the environment and to use basically these membranes to generate gradients of ions, for instance, protons, across the membrane. So these bacteria, they use the trick. So they have basically a system, a light-having protein system, that steals electrons from sulfur hydrogen. That's basically a substance that is, the electrons are not easily available, and then they are energized by the light, so they get energy-rich electrons, and these energy-rich electrons, they can drive a proton pump. So a light would be used to energize electrons taken from anorganic material, not from organic material, from anorganic material now, to energize a proton pump, and this proton pump would be essential to make ATP via these F-type ATPase and to take up material. And that means these organisms would become now dependent on light, but not anymore depending on these organic substances. They can basically use light to drive their energy metabolism. And you see basically now when you have these components, that is a proton-driven pump that already pre-existed in the other bacteria. It was now only newly combined with a system that can harvest light. So nature always used the components that are already there to reassemble. It's like on the level of the lipids we talked about it, on the level of proteins we talked about it. Now we talk even on the level of protein complexes with the same principle again. Then, probably another million years further, cyanobacteria came up. And they basically found another solution to steal electrons. They were able to steal electrons from water. They could split water. That's something we would like to do now. No? You know, this energy problem on the Earth, so you would like to basically make endless fill. So if you would be able to split water, you get oxygen and hydrogen. And hydrogen and oxygen you can burn again and you would have a lot of energy. So these bacteria found a way to split water, which requires quite a lot of energy. So they developed a light harvesting system that is able to use the energy of photons to steal the electrons from water, to split water. As a byproduct, you get oxygen. It's a waste product. That was a disaster, no? For all the other organisms, because oxygen is very aggressive. You notice, for instance, if you have metal, iron, if you wait long enough, it will be oxidized. And you can imagine that organisms that are living in an unorganic atmosphere are now get exposed to oxygen. They don't have any protective mechanism. This oxygen might attack lipids, the double bonds in the lipids. The oxygen might attack the proteins, the amino acids. So for several organisms, at that time, these organisms were not dangerous in a way that they produced the toxic byproduct, oxygen. All this oxygen first accumulated in the water, but at a certain moment the water was saturated and our atmosphere started also to get oxygen. Okay, so these particular organisms were able to actually take electrons from water, to split water, to energize it, to use basically in these electrons to drive again a proton pump, to make a proton gradient. And then they even had a second system that could re-energize the electrons once more to be transferred to the reducing substance. And that is important for metabolic processes. We don't go in detail here. That's more for the biochemist. But they could basically energize the electrons to drive proton transport across the membrane and to re-energize the electrons to give basically new chemicals that are important into metabolic pathways of the cells. So these cells could completely live now on light. Phototrophic organisms. Okay, so now we have basically here these evolutionary steps, and I talked about that now with the appearance of basically cyanobacteria, our atmosphere got oxygen. And that allowed to develop basically another type of cell that could use oxygen. And those cells would have an advantage because those cells would oxidize now organic material very efficiently down to carbon dioxide. Normally, when you do for glycolysis, for instance, you still end up with an energy-rich substance. It's not completely broken down. You have, for instance, glucose, and you end up with pyruvate or lactose or ethanol, depending on the organism. It's still energy-rich. No? I mean, ethanol you can still burn, pyruvate you can still use. But if you can further oxidize it now with the help of oxygen, you get the maximum of energy. Actually, you almost burn it. You burn it in a cold way in the cells of our body, and you can get a lot of energy. So these basically cells here, they would be able to use the oxygen and take electrons from the organic material. They would live from organic material and then transfer these electrons down basically to the oxygen to form water. And while they do this, they would again drive proton pumps to generate a proton gradient that then can be used to make ATP. So in all cases, you have basically always a membrane involved that is required for the basically transmission of the energy of electrons into the later formation of ATP. Okay, So if you now consider this in terms of evolution, so you would basically have here an organism starting as a single cell, and maybe this cell, this organism at a certain moment was able to generate quite a lot of membrane. The more membrane you have, the more processes you can basically let go on, the more proteins you can embed. No? So this organism here already would be a type of cell that would still live from organic material, no photosynthesis, nothing like that, but would have a high capacity of producing material and to break down material due to the large amount of membranes already in. And then an important event happened. So this cell, the so-called pro-eukaryote, took up another bacterium. But instead of eating it away, it made a friendship. Instead of basically using this bacterium and to digest it, it started to make a friendship. And that basically ended up with the first type of heterotrophic cell, a type of precursor of the animal cell that then went to animal cells. So those cells, part of these organisms that were able to basically now harvest the energy or to share the energy that is produced by this bacterium inside, they basically then took up another bacterium, cyanobacterium, that became a plastid. So now they had a chloroplasts. So you have basically now from the original cell that was an organism using only organic substances, you had basically now an heterotrophic eukaryote that was able to produce ATP from organic substance by the use of oxygen, and you would here have a photosynthetic organism that would be able to produce basically ATP by the use of light. So that's the way how basically in the end our cells, the precursors of our cells were formed. And then this cell gave later evolution to all the plants that we know now. This cell gave then rise to all the type of heterotrophic organisms, animals that we know. Okay, so what is common in all these? Common is, it's the membrane. The bacteria, the plants, the animals, they all use the membrane, a bilayer, in which basically a proton pump, a protein is embedded, or a protein complex is embedded, that can use electrons to pump protons across the membrane. That allows to generate a proton gradient, and this proton gradient is then used by a second transporter complex, by a second membrane protein transport to generate ATP. That's common for the mitochondria in our cells, and those two protein transport complexes would be localized on the inner membrane of the mitochondria, and in the chloroplast it would be localized here in these thylakoid membranes. So just to make sure that we are all on the same, so this mitochondria, why would they have the mitochondria two membranes? What is the reason that in ours, basically the mitochondria have two membranes and not only one membrane? Because we would have started with a bacterium that would be have taken up but not digested, but stays in the cell. So where does basically this membrane comes from? The outer membrane? Where does it come from? From?
Outer membrane is from the host cell.
It's from the host cell. It's almost from the plasma membrane. This membrane is a plasma membrane-like, and this is the membrane of the original organism taken up. What would this tell you without much knowledge about the lipid composition? What would you expect for these two membranes in terms of lipid composition? Very different. Very different, because one is a foreign organism that you take up, but you don't digest, but you encapsulate it with your own membrane, which comes from you. So this membrane would be most similar to the host cell. This membrane would be very different. Okay, now this is not maybe well drawn here. You have here the basically chloroplast with the thylakoid membranes. You have an inner membrane and an outer membrane. Why do we have there 3 membranes? Maybe we go back here. Oh, sorry. Now we have an organelle with 3 membranes, quite complex. This is because the starting organism already had 2 membranes, was taken up, engulfed but not eaten up, which means if you think about the membrane, the outer membrane, what would be the lipid composition? What can we expect from the lipid composition of the outer membrane? Would be most of the host cell. What can we expect for the lipid composition of the thylakoid and the inner membrane? They would be more similar to the cell that have been taken up. Yeah, you see? So and indeed it is like that. So if you would basically, and that is actually also support for this endosymbionten theory where people came up with. You can actually do this also on the biochemical level. The membranes are different in terms of outer membrane, inner membrane, for the chloroplasts and for the mitochondria. Okay, good. So now let's have a look a little bit more in details to the membranes. So this would be now the mitochondria. We have here the outer membrane and we have the inner membrane, and you see that this inner membrane is so typically invaginated. This is also what you knew now already from the school when you would draw. The mitochondrium in the drawing is most easily to detect because the inner membrane is quiet. No? And what is the reason for that? You would typically what say, why would this membrane maybe, in evolution be so invaginated? What could be reasons for that? You want to give me some reasons? Surface, that would be the typical answer. Thomas, this is, we need a lot of surface. When we have a lot of surface, we can put a lot of these transporters in the membrane. So we have a very efficient system, right? Let's see whether this is the whole truth. There is some truth in that, but not necessarily all. And we have basically here the chloroplast, and sorry for the drawing, so you have basically the outer membrane, inner membrane, and the thylakoid membrane. Now the machinery that makes basically this gradient and produces ATP is sitting here on these thylakoid membranes. Why would Thomas have drawn this green? Is there any reason to give this a green color? Chlorophyll. So, chlorophyll. Very interesting molecule. And this chlorophyll is really interesting because it's bound to a membrane protein, right? And this membrane protein is then basically pumping protons, right? So it's the light harvesting part, which basically takes the energy of the photons, and this energy of the photons, it's transmitted into the movement of the protein that starts to pump protons. Okay, so in a way, it's not absolutely true. The inside is not green. It's actually the protein that is in the membranes that is green, right? But since these structures are very small, you can just see green, no? And also from the leaves, you just see them green. Okay, so now there's one important aspect. Okay, here we produce basically a proton gradient on one membrane, and we have a membrane that is basically around it. Here we produce the proton gradient in the lumen. So obviously in the evolution, something went differently in the terms of mitochondria and in terms of the chloroplasts. So let's have here a look. I need to tell you that this membrane here, the outer membrane, it's quite permeable. It contains pore proteins, proteins that make really large pores. The consequence would be if you pump protons here into this mitochondrial space, the inner membrane space, the protons have a good chance to leave the mitochondrium into the cytosol. That would be a disaster, right? Because we put so much energy using the energy of electrons to put now all the electrons on that side, and then they disappear. Actually, in fact, this is wonderful here. You can see this is an outer membrane mitochondria. Yeah? A lot of pores. So now we have here basically the inner membrane. Now we pump protons here over with energy-rich electrons. In the case of mitochondria, it comes from organic material. So we pump them all here, and they would start to diffuse. And they would go away. But in fact, we want to have the protons here to be used by this F-type ATPase to leak back and to drive ATP synthesis. So from the construction, this system is not nice. So actually, somehow, as a type of engineer, there's a failure in this. Right? In case of the chloroplast, it's different, the situation. There you pump the protons into a closed lumen. They're all here. And they cannot escape unless they cross the membrane again via an F-type ATPase to make ATP. And due to this problem, this drawing here and this construction is also not absolutely correct because the invaginations are very tight, also very, very thin. Yes, you have a large surface, but what do you see now? You make almost a separation here from the space, the inner space. So all the protons that you pump, they accumulate. Yes, they might have a chance to diffuse, but diffusion is limited. So they stay quite here and can be immediately used by the ATP synthase to generate ATP when they flow back to these proteins. So these invaginations, as one hand is to make a large surface, but the second thing is also to limit the diffusion of the protons that have just been pumped by the electron-driven proton pumps to the basically mitochondrial space. Okay, so these constructions, they have a biological, actually, also physical type of reason to limit the diffusion, to maximize the use of the protons. Okay, so now you have basically seen the basic mechanisms, and we go just a little bit more into the idea of how to use a membrane to make a gradient, because I told you that in evolution, not only one electron-driven proton pump developed, but actually several, and they teamed up. So what you see here is now one proton pump, a second proton pump, and a third proton pump. And they are all in one membrane. And they all use the energy from one electron. So the electron, in this case 2 electrons, are delivered to this proton pump, which, when you have an energy-rich electron, that starts now here to go into this protein complex. And while it is walking to this protein complex, you will change your conformation. And this change of conformation will allow a proton to pump. Now, this electron is still not completely energyless. It can go to the next proton pump, which also triggers now a movement and pumps protons. It's still not lost all of the energy. It still has enough energy to even drive another pump. So you use the energy of 2 electrons very, very efficient by allowing these electrons to pass 3 complexes that all pump protons. So you generate a strong proton gradient by just taking 2 electrons from an organic substance that you break down. Okay? So for historical reasons, they have now not numbered exactly like you would expect 1, 2, 3, but 1, 3, and 4. But this is something for those that really want to dive into the details. The principle is 3 proton pumps. And in between, you see you have also helper proteins or helper molecules that help in the transfer of the electrons. Okay, just since we are talking about membranes and we're talking about membrane proteins, if you want to design this in a real way, not like it's basically there drawn, I think I would like to have your help again. Huh? Yes, both, because we need 3, no? We need 3 persons here. We would be 3 proton pumps. No? We would be very happy, because whenever an electron comes,
you would basically pump a proton. No? And we would do this, pump a proton. The first electron, so the two electrons come to you. What is your movement? Yeah, we are in the membrane, and we basically pump in this direction, make the movement.
Okay, so how is it supposed to be?
Yeah, like this, yes, you are right. Yes, so now we do it, so okay. Electron comes, you, action. Electron comes to me, action. Electron, no, you not. You gave, no. Electron comes to you, okay. You get your new electrons. Then I get it. You get your electron. You get your electron. Okay, but we need helpers, no? Tanmay, you want to be one helper? If you go a little bit more distance, so you would be, for instance, you could be a soluble helper. You are in the membrane and you have this fantastic ability to take electrons. So you can go on his side shortly, take the electron, bring it to me, and then I do this. And then we would have here another helper, and then you would do that. Okay, now we do it stepwise. And the drawing is like that. You are the helper. You can always like this. And you are always busy. Yeah, you are always busy. You walk, and whether you have electrons or not, you walk. Sorry for that, but you walk. You walk. Okay, now go ahead. You get electrons.
Yeah.
First this. Yes, then you give an electron. Zack. And then we have another helper here. Zack. You still walk. Do you still get electrons or today not? Where are your electrons?
Yeah, I have more electrons.
Yeah, you get more electrons. Good. Okay, thanks, Tamar. Stop. Wait. Do you think it's like that? Is this a good way? Because that would be the drawing implying, right? Would this be? Have you watched Tanmay here? Can you pretend as if you would have electrons? Okay, thanks. Would it not cost time? And it's a membrane, right? So if we would have a two-dimensional, what else could you do on movement? So now, walk, pump, get electrons. Yes? How did you know that he has no electron?
I would just try to
Yeah, you would always walk, and this would be not very efficient, right? There would be a delay. Do we want this? We want to be good. We want to be fast, right? What would be a solution? What are solutions you could come up? What do you think? To make this more efficient, this process. Do you have some fantasy? We always need students with fantasy because sometimes, you know. Okay, yeah, that could be an option. But what if, I hope you are not, it's okay with you, Tanmay, and your colleague comes also. Would this be not cleverer if we come closer together? If we come very close together? And would Tanmay need to move no much? What would your movement be for the electrons? Ah, more, faster. Yes, good. What is about you? Do you want to have this? Yeah. So how do we call this now? A protein complex. That would be, so actually, in fact, you know, what this drawing is just to illustrate a little bit that you see there are proteins. But in reality, they are very close to each other. It's a super complex that we are talking about. We are actually sitting so close together that Tanmay as a partner would not need to diffuse your, you know, endless around. You would be in this complex very fast. And you would be also less stressful for you here, no, to walk around. So we would be super efficient in this. And that's what's in the reality is. So these proteins are super, super packed. No? Okay, thanks for that. That was a little illustration that this drawing is actually misleading. It serves a good purpose to explain each step, but it doesn't reflect when you would basically put these proteins in the membrane, they would form a complex.
very tight complex. Okay, good, that we have understood. So now, basically, okay, we could put this in a membrane, we could basically give a substance that delivers the electrons, and we would get proton pumping. So we could build it in a vesicle. And then we could start pumping, basically, protons. So we would then get in the system either an accumulation of protons in the vesicle, or you would move out depending on the orientation, how you have inserted this into the vesicle, right? Yes? Much. There's basically maybe one lipid in between or so. There is hardly any membrane between them, but they sit in a membrane. But the proteins by themselves, they form a super complex. So this drawing, it's more like to understand the steps, but it's not the situation in terms of topology, how the system would really look in the membrane. Okay, so now we have a proton gradient, and what else would we now need to get ATP? Which type of protein would we need to include in this complex? Exactly. So we would need now this type of protein complex that would use the proton gradient to make the ATP. Okay, so now you see how a membrane can be used basically to generate ATP by protein complexes that are on one hand generate a proton gradient, and by a protein complex that uses this proton gradient to make it. Okay, so like very briefly, remember the idea of this transport complex was it works like a turbine. So the protons, basically, that the proton gradient that is generated, the protons would go back through this protein complex, and then one part of the protein would get in rotation. And this rotation would basically affect the conformation of this F1 protein complex. It's in 3 subunits that can bind ADP, phosphate, make the bonding, and you get ATP. And it does it basically in a cycle. You have 3 subunits. Each of them can bind the ADP, the phosphate, and can link it. So in fact, when you have 3 and you have basically now the protons flow, that would give me a rotation. And if I have 3 subunits, we could still play this actually. You want to be a subunit, Tanmay, and you want to be the other subunit. You can turn to this side here. Sorry, you can turn your face to the audience. Yes, like that. Turn a little bit more. Yeah, like, okay. Yes, you can be a subunit like that. You also want to be a subunit? Yes. Then we all move a little bit more like that. So. And you are the other one. So you turn to this side shortly. So what they all can do is they can bind ADP, phosphate, combine it. But they would only do this type of literary movement. No, I don't. So no, I do this, but it's not true. I do this. And then Tanmay can change basically the conformation. No? You do your own job. You make ATP, you make ATP, you make ATP. No? So suck, and I get dizzy, but the transporter doesn't dizzy. And this turning is because of the proton flow here on my legs. The proton flow drives me crazy, while I basically always make these subunits here busy. Okay, thanks a lot. And then we have our ATP. That's what's going on. Good. Now, wonderful, all fine. But what happens? Where do we get the ATP? We get it produced inside. It's all inside of the mitochondria, in the matrix. What a disaster. Because where do we need the ATP? We need it outside. We need it in the cytosol. And ATP is not permeable. Why is it not permeable? Why can ATP not cross a membrane? What were the three things you should then think about? The 3 criteria. When is a substance permeable or not permeable? Size, polarity, charge.
What is it about ATP? Large. Of course, there are larger molecules. Proteins would be larger, but it's larger than an ion. What else? Charged. 4 times negatively charged. Super negative molecule. A super negative molecule, ATP. 4 negative charges. One of the most negative molecules, no, if you think. In biology, at least. In biology. So this would never leave a membrane. It will never cross a membrane. So you need here again a transporter, an exchanger. You see? And this would basically, this exchanger is an antiporter. It allows ATP to go out and ADP to go in, because the ADP is needed to form ATP. No? But what do you see here? What is the charge of ADP? It's -3. That's the charge of ATP -4. What would this mean? If you exchange a substance that is basically 3-fold negatively charged against a substance that is 4-fold negatively charged. There's a charge imbalance, right? It's not equal. It's not minus against minus. It's 3 minus against 4 minus. But in this case, you see the proton gradient. It helps actually to get in the ADP and to get out the ATP. So it's a wonderful system, this antiporter. No? And of course, now you can see you need even other, basically, transporters for other material to go into the mitochondria that would then help to deliver the electrons in the end. No? You would need, for instance, in this case, an organic substance pyruvate that is further broken down, delivers the electrons that are then used by these electron-driven transporters. So this inner membrane, it's basically comes from a very busy organism in the evolutionary time. So it's an organism that was able to generate ATP from a proton gradient using organic material. And this we still carry in us in a way that we became, our cells became friend with this organism. Okay, so this is just a summary here that you see that basically in evolution, nature found similar solutions for a problem. How to make ATP. In both cases, nature uses membranes, a lipid membrane. In both cases, nature uses proton pumps that generates a proton gradient. And those basically gradients are then used to drive an ATP synthase to generate ATP. In the case of the mitochondria, the electrons come from our nutrients that are broken down. And in the case of chloroplasts, the electrons come from the water that is splitted by the help of light. So those organisms can basically produce ATP just from light. Those organisms carrying mitochondria can produce ATP only from organic substance. So how is it now, how would it be in a plant when there's no light? They will use the mitochondria because plants, remember, they have mitochondria and chloroplasts. If there's light, they use the chloroplast. If there's no, in the night, they still need to make ATP. But they can do this with the help of the mitochondria. So part of the substance that they have produced during the day need to be broken down again to sustain, of course, the life of the plant during the night or when there's very dark periods. Yeah, with this, I would like to end. Just to remind you, probably the first membranes were basically used to allow the generation of proton gradients. And these proton gradients were basically the basis to generate ATP and to allow the uptake of substance by so-called secondary different active transporters. And the formation of these membrane structures was basically the prerequisite that you could have an independent organism. And the evolution of the electron transport chain was basically important for efficiently use the energy of electrons to generate a proton gradient. Remember, we had not only one, the early organisms maybe only had one proton-driven, electron-driven proton pump, but then the later organisms, they actually were able to have
a chain of membrane proteins that use electrons to pump. And that we call respiratory chain now, for instance, in the mitochondria of our cells. Yeah, that would be for today for bioenergetics. So we have a very basic introduction into bioenergetics. You have seen how important are membranes in this process. We will then later, after the break, come to a new topic, how are membranes used actually in signaling processes. Any questions for this part? Yes, please.
Why do we need the molecule that needs to transfer the electrons? Why cannot we have all the three forms together in the room something?
That's a good point. So the question is basically, Thomas, why not let's put all these three proteins together? Why would we now need transfer, basically? Actually, I don't know exactly the answer for this question. You probably need to go into the structure of these proteins. It has probably something to do with the place to deliver, the entrance site for the electron. So somehow it's basically that these proteins developed independently, but then joined together. And this joining together, then it was not possible anymore that an electron, basically, that the two sites where the electrons are released and then given to the next proteins, that they overlap. That would be my simple explanation, but I don't know whether this is really the truth. So we would all need to look this up into the literature, why basically in evolution these transfer helpers developed. The easiest thing would be Tanmai was in a bacterium developed, then I was in another, and then somehow, for instance, of exchange of genetic material, we both ended up in the same cell. We ended up in a cell that could produce both of us, but that we would work together would be only basically not possible by directly contacting us, but having a helper molecule. That would be, for me, the easiest explanation. But very interesting. So you make me curious also to go more into the details of that. It's evolutionarily, I think it's very interesting. More questions? Yeah. Yes, please.
So once you say that use of the proton pump, it drives the proton outside the membrane, then is there not the problem of diffusion of those protons before they are being used by that type?
Yeah, that's a very, very good point. So the question is, Thomas, okay, now we have heard that protons have been used very early in the evolution to actually in the generation of gradients. And if you think about a proton, is this a good choice? So what, if you compare, for instance, yesterday we talked about sodium and potassium. And now we talk about protons. What would be the size of sodium, potassium, protons? Not the exact, but relatively. Would you say protons are small? Much smaller, much smaller, much smaller. And then you have basically a lipid bilayer, right? Which we said basically these lipids can move, and also on the other side can move, so you can have basically space in between. Could not protons just pass? And they do. So for protons, actually, membranes are not so tight as we would like to wish, because we have these three problems. No? Protons are small, so they can pass. They are charged, so they cannot pass. But since the size is here very small, they have a higher tendency to pass. So it's actually, strictly speaking, a miracle why nature chooses proton gradients first. It's not clever in terms of the tightness of a membrane. You would rather use sodium or even divalent cations. So that's probably then another type of lecture where we need to invite another expert. What could be the reason that protons were first selected as the ions to generate a gradient? From the type of membrane, it's the most difficult task for an organism to make the membrane tight against protons. It's extremely difficult. And when you do, for instance, these model membrane systems, you have already made giant vesicles. These membranes are quite, so if you change the pH outside, it will take a moment, but
maybe minutes, depending on the lipid composition, sometimes only seconds. Also the inside will change, but definitely minutes. If you would basically look for the permeability of sodium or potassium, it would take hours that they basically pass. Yeah. So evolutionarily, it's actually a disadvantage in terms of having an efficient system to pump protons, but on the other hand, these protons can easily diffuse freely back just by passive diffusion. Yeah. But it somehow still was successful enough. I mean, at least for the plants, you remember they had this proton, they pumped it also in a lumen to basically only have this problem of permeability with the membrane and not the problem of diffusion even out of the organelle. Like in the mitochondria, where you have this problem that the protons that are pumped in the inner membrane space could even leave the mitochondria completely. Yeah. So there's also modeling. So people have done mathematical modeling. So what would be the permeability in protons? And protons are quite mobile across membranes relatively to other ions, which also experimentally makes it difficult for us. If we want to work on these questions, it's technically demanding to generate tight vesicles, and then still the diffusion is fast, to measure then still the diffusion also. There are techniques like fluorescent microscopy on GOVs you can use. You would basically change the pH suddenly and then follow the pH inside of the GOV, and by that can determine the proton permeability. Yeah. Okay, very good question. Okay, then let's have a break. We can, of course, continue to discuss. And who can help me? When should we meet back here for the next session? Would it be 12 or 11? Half an hour now. We still have time. Half an hour. Please enjoy, and let's join for the tea. Thanks. Anyone missing the colleague? Or all here? Yeah, looks good. Good. Okay. We still wait. I can see some people are probably still having a little bit tea outside. We still have 3 minutes, so please continue to talk to your neighbors, no problem, and then we start sharp in time.
Okay, so maybe I can talk to you for 2 minutes. So how many of you are not PhD students? I mean, who are registered. Who are registered for the course but not PhD.
There are 2 more, but one is at campus and one is outside.
3
Yes, ma'am.
and 4. 4. So for PhDs, I'm assuming that this registration slip that you're paying, no? For all of you, you can claim it when from your residency or somewhere, no? You'll get it back, right? So I just wanted to check with you. Not so I just wanted to check with all the PhDs, so you can fill up the TA form and get your accommodation. You can claim it. Is that correct? What is the maximum you are allowed to take for the? There an upper limit, I think it is there, no? So one of you told me that it is not allowed for you to take 900 per day. So what is allowed? 400 allowed? 500 allowed? Any number so that then I take care of it rest of it. How much you will not get back is what I'm asking. I'm just trying to help you. But 450 I think you can, no? 450 per day should be fine. Yeah, but I also don't know. So that is so 450 is fine per night, right? And food expenses are not too much. No? So forget about the food. I take care of the food. So accommodation 450 should be fine. No? Right? Everyone? If anyone has a problem, you do let me know. Okay? And for those who are not PhDs, what do you claim anything back? So you please talk to me separately. How many of you are there? 3 or 4?
Yes, ma'am.
You talk to me separately, please. All those stalls being or not being. Okay, don't forget. Okay, let's start the lecture now.
So welcome back to the next lecture of the day. We are now going to one other aspects where cellular membranes are very important, and that concerns signaling. In fact, the membrane, in this case, it's a dilemma, right? If you want to receive signals from the outside, or you want to give information to the outside, you have a barrier. But on the other hand, if you make this membrane selective for receiving or giving signals, you have a type of filter. So only those information that maybe for this cell is relevant is then really able to cross the membrane and would, for instance, induce a cellular change, right? So let's have a look how this is realized in the context of organisms. So let's start with us as a type of multicellular organism, right? So our whole body is composed of over 200 different cell types, nerve cells, heart cells, liver cells, and some of them need to do cell division. Others need to grow, or cells need to differentiate. Some cells need to die. So in such a complex organism like us, where we have this enormous amount of different cells, it's only possible to survive when the cells start to communicate with each other. So without basically communication, multicellular organisms, it's not possible. In particular, specialization of a cell is not possible. So these cells that we have, they are very specialized. Those cells are important for signaling communication between them. Those cells are important to have basically the muscle construction of the heart. So each of the cells, very specialized. Liver cells are a type of chemistry fabric in our body. They are very specialized of breaking down material and making material. So definitely without cell signaling, no multicellular organism would be possible. No specialization would be possible. And the signaling allows the coordination of cell division, growth, differentiation. It allows the fight against pathogens to the outside. And it's important for sensoric and neural processes. Makes specialization possible. But most likely, this started earlier, already on the unicellular level. So already bacteria actually are able to talk with each other to signal. One example would be the biofilms. So maybe you have seen on lakes, depending also from the weather conditions, you might get actually a type of thin layer, which often are bacteria that start to aggregate. And that's sometimes for a particular period when in the year when they are basically well growing, and for particular conditions of nutrients, they start to form these biofilms. So already bacteria actually need to talk. So what we need to understand is how do they talk? What is the language of cells? How does this work? So first of all, basically, cells talk with the help of molecules that are basically released or accepted by them. And you see here just the diversity of molecules that can actually serve as signal. In our body, growth hormones or insulin, you know that insulin is, for instance, an important factor in our body for the regulation of the blood sugar level. Insulin would trigger the uptake of glucose from the blood into each of our cells. So obviously our cells recognize insulin as a signal to take up glucose. What would we require to take up glucose? Do you remember? What would be important then in the cell? Because we have a membrane, glucose, not very well permeable. So we would need a transporter, a glucose transporter. So this glucose transporter would be then basically taken to the plasma membrane in a response to insulin so that the cells are able to take up the glucose and can use it for their metabolism. You see basically that you also have type of lipids. That's a steroid. Lipids can be signaling molecules. No? Or you see like small chemical molecules here that are more derived from an amino acid, like adrenaline can serve. But even such small molecules like gases, even gas can be as a signaling molecule. That's astonishing. No? So what would be the structure that recognizes such a small gas molecule as a signal? That was puzzling over many, many years before people basically found a protein that is able to bind this gas and to actually use this as a signal. So you have quite a diversity, and now it depends, of course, from the type of cell, from the type of organism. What other signals can you think about? What about bananas? I had here two helpers that were so kind to actually buy some bananas for me. And if you let these bananas lying, what happens to these bananas? They turn yellow, no? Do you also maybe know from your parents that the parents would say, don't put the new bananas close to the old bananas? What is the reason?
They influence the other bananas to ripe faster.
The other bananas ripe no faster. What's going on? They talk with each other. The bananas that are old talk to the bananas that are new. And they do this via a gas, ethylene. And that gives this nice smell when you have in a house and you have these bananas that are very mature. You can actually smell this ethylene almost. So bananas talk. Or maybe you have heard about butterflies that can receive signals, pheromones, over kilometers. Can you imagine? Very few molecules, chemical molecules, that are recognized by a type of structure in these butterflies as a signal. Amazing. Very low concentrations, very, very sensitive sensors that can do this. So nature uses a lot of different type of molecules to serve as signals, and it depends, of course. We cannot detect these pheromones because we don't have the machinery to detect them, but in the butterflies, and some of the butterflies, they can recognize, and they can, of course, only recognize from their own species these type. No? Okay, so we call basically these signaling molecules that are exchanged between cells so-called first messengers. And you will see the reason why we call them first messenger later. So in all cases, basically, we have here the cell surrounded by the plasma membrane, and typically it's a membrane protein that can bind and recognize this first messenger. And when it does so, it will change the conformation. So maybe like click. So it will change the conformation. And that typically leads even the change of within the whole protein that gives then a possibility to the cell to recognize something has bound to the outside because the inside has changed. And now basically this change triggers an intracellular cascade. So now comes other molecules that are activated and transmit the signal, and that can basically then end up to molecules that, for instance, change gene expression. So for those that are more in biology, it would be proteins that can bind to the DNA and either activate the gene expression or not. We call them transcription factors. Or it could be an enzyme that is involved in metabolism to produce, for instance, a particular new molecule that is required. Or it could be a protein of the cytoskeleton that will change now the arrangement, and it will change the shape of the cell. So that we call basically the effector proteins that would be then change their activity depending whether a signal has bound to the outside or not. And what you also see is basically that since you have this type of cascade, you have here the possibility of an amplification. We will come to this a little bit later. Okay, so now, very simple. You of course, are welcome to dive even more than if you are fascinating about signaling into this topic. Let's have a look on these proteins that serve as receptors. And you can group them very, very roughly. It's very rough. You can group them in proteins that look like that. We call them G protein coupled receptors. This would be proteins that actually interact with another protein, which is a G protein. It could be these type of receptors that carry an enzyme activity. So those receptors by themselves are enzymes that can be switched on and switched off. That would be a receptor tyrosine kinase. Or it can be basically proteins that help to recruit other proteins, adapter proteins. Okay? It can be an ion channel. We talked about this yesterday. An ion channel can bind the ligand, open or close, depending on what type of ion channel we are talking about. And a family of receptors we are not going to talk about today would be, for instance, receptors that are intracellular. But those would normally only recognize a signal when this signal can cross the membrane. So what do you think? What molecules would be able to cross the membrane? Small molecules. No? And if we go back here, sorry, from this basically here type, no? This would be a lipid-like molecule, right? And if it's lipid-like and maybe uncharged, it can also do flip-flop, and it could go in. So steroids are relatively easy to flip-flop, steroids. They have this very small head group, very large hydrophobic backbone, this steroid. And that's why steroid hormones, they are basically able to cross the membrane without help. And that's why the receptor can be inside. So that's the reason why for some cases you can have basically the receptor inside. But what we want to discuss is this family of receptors here on the plasma membrane. And we will only focus on 2 examples, just that you get an idea of how do they work. Okay, so one other important aspect is we have not talked about the molecules on the plasma membrane, the receptors. They can be grouped in different families. The other thing is that inside of the cell you have molecules that are, we call them molecular switches, almost like the light. They can be active or inactive. And there's 2 families of those proteins that work as switches. There are proteins that can be phosphorylated and dephosphorylated. And we have already talked about phosphorylation. It's always a drastic event because you transfer a charged molecule, phosphoric acid. And this type, then the protein becomes the area where it has phosphorylated, 2-fold negative. And you can remove it. And the protein that control these phosphorylation, dephosphorylation are kinases and phosphatases. So you have here a switch that is off when it's not phosphorylated, on when it's phosphorylated. And you can control this by a kinase that is phosphorylating and a phosphatate that is dephosphorylated. So you can switch between an on and off state. The other family, it's the so-called GTP binding proteins. So these are proteins that can bind the molecule and can change this molecule, and that switch between on and off. Let's go a little bit more in detail. So let's start with these G proteins. So these G proteins, they can bind a molecule that is called GDP, and they can bind GTP. So typically they bind GTP, and they have the ability to change this GTP into GDP. When they bind GTP, which is basically analogon to ATP, then they are active. When they basically hydrolyze this GTP to GDP, then they are inactive. So again, they can switch between an on state and, how do we say, GDP, phosphate, on state, off state. Then, of course, this molecule needs to leave, the GDP needs to leave. Now comes new GDP in, on, off. Okay, that it can do. It needs eventually to change from one condition to the other, also helpers. It needs basically helper proteins to get into the on state, to exchange the GDP against the GTP. And it needs basically a helper protein to be activated to hydrolyze the GDP to GDP. Okay? And these helper proteins, these are basically here just given the GTP exchange factor and a GTPase activating protein. You don't need to remember this now for this course. It's only for those that are very interested in. Important is that we have a switch that can change between 2 conditions, and this switch is controlled again by helper proteins. There's even another helper protein that can move these G proteins in an inactive state. Okay, so what we are doing is basically we have at the plasma membrane receptors that can bind the signal, they can change the conformation, and that triggers here an event in the cytosol. It activates switch proteins. It can basically activate switch proteins that are phosphorylated, dephosphorylated, or it can activate switch proteins that are binding GTP and then hydrolyze the GTP to GDP to get inactive. Okay. So here's this example for the phosphorylation. Protein that is in the off state gets phosphorylated in the on state, a molecular switch that can then be switched back to the off state by removing the phosphate on the protein. Okay, now comes one thing more. These phosphorylation steps can also be coupled. Before I basically go to this, it's maybe easier to play. The point could be the following. I would be basically now a protein, that gets phosphorylated. But I by myself have the ability to phosphorylate now another protein, meaning I would be a kinase. And Tanmay already sees that I look in his eyes to come here. Okay, so Tanmay could be just another protein, but he is my target. If I get phosphorylated, I could be a kinase now, that gets activated by this phosphorylation, and Tanmay I would bind ATP and transfer the ATP to Tanmay. Then he could be active. But what is if he would be now another kinase that gets active? Then you would come into play because you are the target of Tanmay, and Tanmay would activate you. So now we need even another box. Tanmay activates you and transfers the phosphate to you. And what is when you are also now a kinase? But in the moment, we only have done 1 to 1 to 1. Now, Tanmay, we have several copies of Tanmay. Do you want to be Tanmay second? So you have to imagine this is now Tanmay again. Stay here. Okay. So I'm a protein that gets activated by phosphorylation. I have the ability now to phosphorylate other proteins. These are my two targets there, Tanmay, Tanmay. So I would basically phosphorylate you. I would phosphorylate you. Now only one protein has activated two proteins. Now you both are activated and would have 10 times, what was your name? Hindon. 10 times. So then you could start to activate, you could start to activate. You see, so you get an amplification because what is drawn there, it's a little bit misleading because there's always several copies of the same protein. Maybe Thomas is existing 3 times. Tanmay is existing 10 times. Hydron is existing 100 times. And all 100 molecules could be phosphorylated only by 1 or by 2 or by 3 of those guys. You see? So it is as if basically your target would be all the others, and you would activate now all the others. Okay, and we call this a phosphorylation cascade. A phosphorylation cascade that allows to activate not only one by one, but that one molecule can activate several thousands of the substrate that are present, and so on. Okay, thanks a lot for that. Put this here.
In an exponential pattern?
The question was, Thomas, can this be in an exponential pattern? No? In principle, yes, because it's determined by the number. Now we have here a number of people that can actually do nice modeling. No, you could do now a modeling. You could say, okay, I have 10 copies of this, basically, of this molecule. Let's have 10 of those. No, how would be the signaling then? Or do I have 10, 100, and I take here 1,000 of those molecules? How would be? And if you have enough, basically, then it would be exponential. Then it would, no? It's not always realized like that, but in principle, yes. That would be a very fast amplification of the signal in an exponential way. Okay. So now, yes, please.
Where would the energy for the amplification come from? Obviously, you're increasing the energy, right?
Yes,
What
yes.
is consumed?
Very good point. The energy comes from the ATP, right? So it's stored in the ATP, and whenever you basically now use the ATP to transfer the gamma phosphate, you hydrolyze the bonding. That was what Tripta explained yesterday. And this gives a lot of energy that allows a new bonding to be formed. So it's from the ATP. Yeah, that's important. Okay, so that would be this cascade. So you would have eventually only one receptor activated. But then you can, in the end, that results to an activation of hundreds or thousands of copies of the same protein inside. Then another important aspect is that cells inside also use signals, so-called intracellular signals, second messengers. And those can be of very different flavor. It can be membrane-anchored second messengers that are produced after a signaling process, or it could be soluble second messengers. So calcium, for example, we have learned it's a lot of calcium outside because our cells outside have basically the type of ocean water composition around. But intracellular, in the cytosol, the calcium level are kept very, very low. If you now open a calcium channel and the cytosol is flooded with calcium, the cell definitely sees a change. In particular to those proteins that would be able to bind calcium. If you have no calcium, the protein would be in one conformation. If no calcium comes in and it can bind it, a change in the conformation of this protein would change the activating status. So that's basically molecular switches that are used based off phosphorylation, dephosphorylation, based off binding of GTP, G proteins, and intracellular second messengers are used for the processing of the signals in the cell. Okay, now we have basically one point. We have now talked about the molecules, like the receptor and intracellular molecules. Now, of course, signaling occurs eventually on many receptors because the cells receive many signals at the same time. If basically two signals are received by a cell that are recognized by the two receptors that are present in this cell, it will lead to an event, a cellular signaling event inside of the cell. But it can cause a problem. We can also play a little game. I will say a sentence to you, and you transmit this sentence to the next people, okay? Yesterday was a cold day with bad weather. So I gave a signal. We could make it in the cell, it has to be fast, so we can make it this game now very fast and stressful. Now you need to be faster, faster, faster. Yes, fast, fast. And we have some noise talking also eventually from the outside. It gets now more difficult because there comes a signal from another side. There could be a signal here. I need to go home. And this goes now there. And where's the other signal going here still? It's not going anymore. What's wrong here? Does anyone, did you receive any signal there?
Yeah, but not
What was it?
clearly.
Try the best. What did you understand? It was a cold day with bad weather, was the sentence, and this ended up. Oh, what went wrong here in this cellular communication? That should not happen in the cell, right? But can you imagine now? And then you get another receptor telling also another message. And we have not talked about really the cytosol is full of proteins, right? When you did these GUVs, they were just in a way empty. No? There was only buffer in. But in reality, even now you are not on the level of a cell yet. We would need to invite probably 100 more people here in this room. And then it gets really crowded. And then if I give a message, it will be very hard to give this message for Harshmit, right? Because maybe you need to find Harshmit, no? How do you do this when this is your 100 people in? Maybe you do this here on the Color Festival or whatever. Maybe you also want to find your friends. Now you have the mobile phone, of course. You try to find them, but sometimes it might be very hard. So so basically there can things go wrong. And the cell needs, of course, to avoid this. And there are some tricks cells can do this. So this is what I want to here illustrate. You can basically combine the signaling molecules that should be activated by a helper protein, by a scaffold. So you keep the partners together that should start to communicate, right? Also here, she was not sure what message she should actually receive. Yeah? Eventually there came the message also from that side. So let's say, for instance, in some cases, cells in our body need to decide, whether they should live or die. If you remember, for instance, our fingers, how would this look like in a baby when it would be still unborn? At a certain moment, there would be here still cells. It would be actually a clump of cells. And those cells here, they decided to die. They say, I will die. But such a decision, of course, you should be very carefully decided. Because if you decide here to die, there's no way back. These cells are dead. So then it becomes very important that the cell makes the right decision. So like in your life, when you want to make a decision, do you sometimes need to make decisions? Normally, how do you do this? When you need to make a decision, you sit in your room and then, what should I do? How do you do this? It can be that you just say, do you do it like that? Okay. Okay, I tell you what I do. When I was young, I had a good relationship and still have a good relationship to my family. I went to my father and I explained to him. Then I went to my mother. I didn't talk to them to father. No. My mother said, no, I think you could do this and that. And the father, no, I think. And then I thought, also by myself. So you basically get input from several persons. Can be also your best friend or whatever. And then you make a decision. And say, do the same. If you have a critical decision like life or death, you don't want that, someone tells you, you're on the place to be, no, you don't believe this at the first instant. You need another signal which also says the same. And then you might say, okay, obviously I'm at a place where it's important now that I should be removed. And this is the same. So you have here basically a signaling, intracellular signaling molecule that needs 2 events of phosphorylation before it is really activated. It's not enough just here to activate Tanmai with 1 phosphate. He would be not active. He would need another phosphorylation event, and now you would be active, and now you would continue to signal to other partner molecules. No? Okay, so another point is, of course, very briefly, a decision like life and death, it's basically only 2. There's nothing in between. There's not half life living. No? So how do you turn basically an event into something all or nothing? Because when you have receptors at the surface and signaling molecules bind, one receptor is activated, the other copy of a receptor is activated, and another, and you get more and more activation. But how do you move now this stepwise activation into an all or nothing decision? Those that are very familiar with mathematics, what would it be to go from something gradual to something drastic? It's a type of step function. Step function that you need. So how do you turn something into a step function? No? And one way would be, for instance, you have this type of positive feedback loops, a self-amplification of the signal. So already a small change then triggers basically extreme, like an exponential increase of the signal to get these extreme fast response. Yeah? So they are basically more complex interplays of the molecules to allow gradual changes, but also to decide to allow all or nothing decisions. Okay, good. So now let's take the time for 2 examples. Just it's more like to get a flavor. Let's talk here about this first receptor family. So you see it's a membrane protein. Remember, it was a polytopic here, no? Polytopic membrane several times. And in the case of this G protein, it's exactly 7 transmembrane domains. 7 transmembrane domains that span the membrane. And this protein can interact here with another protein that we call a G protein because it has the ability to bind GTP. So here's the receptor. And you see it has an extracellular part, a membrane-embedded part, and an intracellular part. This part here, the extracellular part, it's typically used to bind the ligands. And this part is used to interact with the G protein. And you see that in reality, of course, you have a 3-dimensional structure. So these 7 membrane domains are basically arranged in a way to get a very compact molecule that has here a binding site for the signaling molecule that comes from the outside, and here the binding site for the interacting protein, the G protein. And depending on what subtype of receptor of this class you have, the ligand binding pocket might be located here, might be located there, might outside or here. And that basically is depending now, if you have a receptor that recognizes monoamines, then it's more receptor where the binding pocket is like that. If it's a peptide hormone, the binding pocket is more located like that. If it's a hormone receptor, often the binding pocket is located like that. And then you have, for instance, here even receptors for glutamate. So you have the same, always the same type of receptor, the same building principle, but they can have different ligands. One receptor of this class might have the binding pocket close to the membrane and would bind the monoamines. Another member of this would have the binding site actually on a loop, on an extracellular loop. So that's why these type of receptors can have very diverse ligands to bind. And it depends now from the cell which receptor would be present, whether they react to one particular hormone or not, or to one particular stimulus. Okay, so now we keep it simple. We say this is our receptor that can bind a ligand, and that leads to the interaction with this G protein. And you see that this protein, again, is composed of subunits. That becomes now complicated. Now we have basically a receptor. This receptor interacts with a protein that by itself consists of 3 subunits, alpha, beta, gamma. And then you see that the alpha unit is linked via a fatty acid, and the gamma unit is linked via a fatty acid to the membrane. So how would we call these type of proteins? We call them peripheral proteins, because they are only not integrated, but linked to the membrane, attached to the membrane by this anchor. Okay, now this unit here, the alpha subunit, it's able to bind GTP in the exchange of GDP. And it would do so when the receptor gets activated. So let's have a look here what happens. So the first thing is, basically we have our inactive receptor, and we have the G protein. You 3 want to come to play again? Okay, I see you want to be the G proteins, the G protein here. You want to be the alpha subunit? So Tanmay is the alpha subunit. What special property does he have? He can bind GTP or not. In the moment you are having not GTP, but GDP. So you are beta gamma, and in principle you can clump together, you are together. Yeah, that's okay, fine. What else does Tanmay have? He has an anchor, a lipid anchor. So you would need to have one hand for the moment up. Don't worry, not the whole, but then we have the beta gamma subunit. What would be the property of the gamma subunit? Having a fatty acid, finger up. So you are anchored. And you are in a close relationship. So yes, exactly. You will always carry him with you, exactly. So now, okay, this protein, so what would this protein, if we do this now for the model membrane, if we would purify, this is a complex, 3 proteins, they would stick together, as long as we have here our GDP bond. What would these proteins be able to do in a membrane? They would move. Can you move? But you need to move together, no? All 3. Why can they move? Yeah. And don't forget, it's a random move. You are not doing random move in the moment. Now it becomes better. Yes, you can imagine as if you would be not knowing where to walk. Okay, why they are mobile? They only have the fatty acid in a lipid bilayer, meaning the lipid bilayer is fluid. Like lipids, they can move like a lipid, but they do it on the surface of the membrane. They would move. Okay, so I would be now the receptor. What would I be able to do? Also move, right? Oh. Sometimes I could come to you, sometimes not. Sometimes I can come to you, right? And they would also move. Okay, so that's nothing spectacular. Until the moment I would bind a ligand. Let's bind no ligand. Now I will be a helper protein for those guys here. I would activate Tanmai. And that would lead to the fact that he would exchange the GDP that he had against GTP. Now he is active, and what happens is he changes his conformation and can leave. Exactly, and now you can walk, and you can walk. So the complex has dissociated into two subunits, alpha, beta, gamma. And they can be basically now separated. So they can do things. They can, for instance, go to those guys there. And you could go here to this side to interact with another person, right? And that would stay like that, but in this case, for these proteins, Tanmai has the activity to hydrolyze this GTP to GDP. Okay? It's now difficult to show it as an example, but he can basically eat up the phosphate, and he will do this slowly. So once the while, one of these alpha subunits would basically, the enzymatic activity would eat up, and then now you have GDP. And that would change, yes, that changes conformation, which when these guys meet him again, makes him sticky to those guys again. So, no? So now they will be in the resting state again. But I still have my ligand bound. So what will happen now again?
The ligand will release.
No, could be, but I'm in the moment, yeah, could be. That would be then the next step. But as long as I have the ligand, what is then?
You can activate it or activate it.
I will activate you again. What will then happen?
Change my conformation and move away from it.
So as long as I have my ligand, I will always reactivate. So what happens is basically now you slowly, now after a while, now is a while gone, what happens? You destroy this, and that gives you GDP. What happens then?
Conformation change.
So the G protein resets itself. So it will be activated and goes into the inactivated, but then they will meet me again, and as long as I am as the receptor, I'm the so-called GEF, the GDP exchange factor, the helper protein, as long as I activate Tanmai again, the G protein will be again basically activated, fall apart. That is what's happening. And only, you are right, when the ligand will dissociate and you will meet me again, then I cannot reactivate them anymore. So that's the principle of basically a receptor that works with a G protein, a protein that is able to bind GTP and has the ability to hydrolyze, but it does it basically only after the while. We call it a slow hydrolysis. Of course, if you have only one molecule, the hydrolysis by itself, it's fast, but it will not happen instantly. It will happen after a delay. Okay? Okay, thanks for that in the moment. And this is a little bit what is now here illustrated on this drawing. So you can go at home once more. You have basically here the inactivated complex with the alpha, beta, and gamma bound with the GDP. Now the ligand binds this receptor, activates the exchange of GDP to GTP, and that triggers the separation of the two subunits of this complex of this G protein into two individual molecules. Okay? And that's basically here also according to this list. The GDP is replaced by GDP. The activated beta-gamma complex dissociates. And then basically the G-alpha, so the alpha subunit and the beta-gamma subunit, they can now interact with other proteins. And then the point is that the G protein can reset itself, but as long as the receptor is active, it will be reactivated. And that goes basically as a cycle. So now what target molecules? I give you just one example. A target molecule could be another membrane protein. In this case, it's an enzyme, an adenylyl cyclase. And when this alpha subunit binds to this enzyme gets active and turns basically ATP into cyclic ATP. It's actually from the drawing here something missing. It should be another bound. So it goes from ATP to cyclic AMP. And this cyclic AMP would be a new water-soluble molecule that would flood in the cell and would have a target protein, for instance, a transcription factor that then can activate gene expression. So from that, you get a signal outside that says this triggers cell division. When this signal comes, could be a type of growth hormone. The cells get the signal, turn on gene expression, produce proteins. Okay. So here's the example. The cyclic AMP binds to, not directly to the transcription factor, but to a protein, which is a kinase. And here's one aspect that I want to discuss just shortly with you. You see that this kinase, again, is actually built up by subunits, two identical subunits, a catalytic subunit that would be able to phosphorylate proteins, and a regulatory subunit. And this regulatory subunit would, as long as it's bound, would keep the catalytic subunit silent. And only when the regulatory subunit dissociates, you get the catalytic subunit active, and that would then phosphorylate the protein, transcription factor, that gives transcription. But there's one interesting thing. You see that basically the binding here, the activation of the protein, the activation of the regulatory subunits requires the binding of 4 cyclic AMP. What does that mean? Let's say you would have here regulatory subunit, 2 binding sites, regulatory subunit. The cells produce 1 cyclic AMP, nothing happens. Now the next cyclic AMP is produced, still nothing happens. Now the next cyclic AMP is produced, nothing happens. Only when the 4th cyclic AMP, then you get activation. So you need a threshold of concentration. For those that are thinking about mathematics, if you need 4 to get the activation, when you have this threshold, that's actually a prerequisite for also these nice step functions. So you need this cooperativity. It's not enough 1. If the cell by accident makes a little cyclic, 1 molecule of cyclic AMP, you need really 4. So the cell really needs to be flooded with cyclic AMP that all the protein kinases that are depending on 4 molecules of cyclic AMP to be activated are really activated. Okay? So it's a type of, if you want to say, sensor system. Whether there's enough concentration, this system would not be linear. It would not react linear with the concentration increase. No? Okay. And this example you find actually in signaling at many places. You need not only 1 copy of the signaling molecules, you need several to get the next step activated. Okay, and then this was basically, now we can here phosphorylate another protein, for instance, a protein that interacts with the DNA and tells basically the polymerase here to make the messenger RNA so that you start protein synthesis. Okay, so now we have basically just talked a little bit on these G protein coupled receptors. Let's take this class here, those receptors that are basically having an enzymatic activity, okay? Now, you see already that they look a bit different from the topology. Obviously, those have 2 single-spanning transmembrane domains, okay? And in fact, they might be in the membrane as 2 proteins. And only when they come together, they would basically get the enzymatic activity, meaning since they are kinases, they would be able to phosphorylate, and they could actually phosphorylate themselves, cross-phosphorylate. So why is this an important step? Or maybe before we come to this, just here for the chemists among us, so that would be here our, for instance, tyrosine kinase receptor, and then you see there's a part of the tyrosine kinase receptor that contains the amino acid tyrosine. And this amino acid can be basically phosphorylated, and in the tyrosine kinase receptors, this is a major event. So when this receptor gets activated, the receptor itself gets phosphorylated on a tyrosine, and that's why they carry the name. And you see, when you do this, this would be polar uncharged, this would be polar supercharged. So you drastically change in the protein itself, again, a part that would be highly charged would, due to the interactions with the other part of the protein, lead to a conformational change. Okay. So you would basically have then the possibility by the phosphorylation of this receptor that you get a change in the environment, in the intracellular environment, that serves as a signal for other proteins to dock. So you create a docking site. And basically this autophosphorylation, if you want to say the transphosphorylation within the receptor, creates docking sites. And the receptor by itself, after having docked this protein, can also phosphorylate the target, so that on top, the target eventually gets, while it has docked, activated. So you have two tasks of this phosphorylation event. One task is that the receptor gets activated serving as a docking, and the second task is that the receptor itself can activate by direct phosphorylation of the substrate and thereby activate the substrate. Okay. Before we come to the two examples, there's one aspect that I want to discuss with you. Tanmay, you want to come? So Tanmay and me, we are the receptor. We carrying here the activity to be able to phosphorylate other proteins, the kinase activity. And normally we would be basically just as monomers. Okay. So now you have to imagine this is the part that is outside of the membrane, this is the part that is inside in the membrane. And I would be only a single spending membrane protein. Let me see. Maybe I can use this. No, this here. So what would I do as a single spending membrane protein? What can I do in the membrane? If you think about the membrane again, now let's go back to the fluidity of a membrane and so on. What can this membrane protein do? I can do this, of course. I can do this. I can do this. All these movements you can do. And I can, of course, in case of a receptor, I can bind here something. What do you think would this mean for the part that is now intracellular? So now I have not bound anything on that part, and I move, and I bind here something, and I move. What would this mean for the inside part of this protein? You think so? It's looks like I look like that. Here has something changed. But if you are so flexible, you know, remember this would be more like, you can hardly do it. If you are so flexible, there will be hardly any change. We can discuss, of course, you know, do I change my diffusion speed now because I'm heavier? But it's, how much change is it? And so it will be very hard to see for the cell inside that something has changed. But what if this binding would trigger something like that? Does then has something changed inside? A complex has formed. That can be seen from the inside, right? I'm alone. Tanmay is alone. Now I bind and you bind ligand. Now we come. That can be seen. And now on top we have an enzymatic activity. I phosphorylate him, he phosphorylates me. And that attracts now adapter proteins. So, thanks a lot. A single spending membrane protein cannot signal alone. It needs to form a dimer. Okay? So these receptors, this is in contrast to these G protein coupled receptors. Remember, they had 7 transmembrane domains. They were quite complex. So there were 7 transmembrane domains. If you bind something, that will change their arrangement between. But with a single membrane spanning protein, there's not, if I bind something or not, there's hardly any change. The fusion rate may be a bit heavier, but typically the ligands are also very small. So then you need this drastic event of dimerization. So these tyrosine kinase receptors, they need to dimerize. That's the only way how they can signal, because they are single spending, no? Biotopic membrane proteins. Okay, so, but there's now 2 things, 2 principles even within in this class. So we can talk about a so-called epidermal growth factor. That's a receptor that tells the cell, you should divide. And we can talk about the insulin receptor. That's the receptor that tells the cell, send glucose transporters to the plasma membrane, take up glucose. Huh? Okay. So in the case of the EGFR receptor, we have this example that we just were dancing. We have basically 2 single spending transmembrane copies that need to dimerize. And the dimerization is triggered by the binding of the ligand. And this basically then triggers the activity of the enzymatic part, the intracellular part, for the phosphorylation. And by that way, you get the receptor activated. Now the receptor is activated. Okay? And in former times, let me see. Yeah, in former times, you see this still in old books, because we had not the structure of this receptor. There was one idea about how they come together. Tanmay, I'm sorry, you want to play again? Okay. So, so what do you think? If you now add to S a ligand, what could be the reason that we come together? I mean, anyway, once in a while we come together, right? Because we diffuse. Once in a while, but this will not keep us together. What do you can come up with ideas? I just want to trigger you, that because that helps you a little bit when you think about problems. So the problem is to understand how can the binding of a ligand keep us then together? Do you want to come up? You can take a moment time. We play again. You also moving still. No? We are as a dynamic membrane. The others can still think. Once in a while we meet, but there's no reason for us to stay together. No? So why would a ligand binding now trigger us to stay together? Okay, one thing that people came up, could it be, because we have 2 binding sites, no? Just look a little bit around here. Maybe, yeah, Tanmay here. Could it not be like that? How do we call it? The ligand cross-links us. That is still what you see in books often. They draw it like that. This ligand binds on this side, no? And on that side. Could also be because the binding is identical, we are identical, no? And now we are together. That would be, and that could be a good idea. And you only know when you know the structure of the complex with the ligand. Only then you can tell. And when you have the structure, then you see that this model is wrong. That is not how it's working. Because I need to bind, you need to bind, but our binding sites are not the sites where we bind together. So when you bind and I bind, we change basically this part here, the extracellular part, a bit. And actually, I need your back. If you turn your back to me, yes, that is fine. And actually, Tanmay already says like that. He binds the ligand and he does this a little bit. And I have the ligand and I do this a bit, and then we come like that together. It's a complete different picture, right? Instead of actually binding like that. This would be cross-linking. The ligand cross-links us. But what the ligand does, it triggers a conformational change that leads to a stickness of us like that. And now we are still together. It's the same principle. We are together as a dimer, and we can start signaling. But of course, this type of mode, you can only find out when you have the structure. But you can come up anyway with ideas, and then you can think about also how to test these ideas. No? Thanks a lot, Hermann. Super. Okay. So that's why also there is a other class of this. They are already cross-linked. They are cross-linked. So we are already forming a complex. And if this is the case, now I don't take Tanmay again, it gets stressed. Can one of you colleagues come to me again? Yeah? Yes? So we would be always together. So like that, we would be always together. But once a ligand binds, we could change, no, like that. So a little bit like that. So and then here these domains come together. So and then we can still form. Because then if two parts are together, we can change relatively to each other the positions a bit. So we can, between us there can be a change. And that can be recognized. So in this case, the problem is basically already partially solved by forming a dimer, a preformed dimer that only needs to change the conformation. In the other case, we need to form first the dimer before we get the signaling. Super. That was the important point that I wanted to make, that you get an understanding basically how the change of protein complex formation and disassembly can actually change a signaling in the process in the cell. Good, not to go, this is really now, we talked about basically, then we have this phosphorylation that helps other proteins to get basically associated. And among those proteins can be again proteins that activate another subclass of G proteins. So that would be a G protein that also is able to bind GTP, and that would activate now kinases, a type of phosphorylation cascade. So one protein phosphorylated